Patty's first, and he'll be coming up right now.
Thank you, Ed, and good morning, everybody. So I'm going to talk to you about the relationship between exercise and affective responses, and I will try to propose a psychobiological model. Ken Fox, in a review that was published a few years ago, summarized what is now believed about this relationship when he said that both survey and experimental research provide support for the well-publicized statement that exercise makes you feel good. Having said that, William Morgan said that, well, it's dark. We have seen this in several studies, and it's now time to move on. He said there is no need for further study or reviews dealing with the question of whether or not physical activity results in improved mood. There are, however, many questions that remain unanswered, and these questions will hopefully be addressed in the decade ahead. He said that in 1997. Well, if you look at the patterns in this area of research, the one theme that is becoming more and more dominant is the theme of dose response. And this is a graph showing the number of studies examining the relationship between the intensity of exercise and affective responses. And the total number now is between 55 and 60. Together with Steve Petruzello, who is going to be our second speaker today, we reviewed the literature up to 1998, and we found that there are 4+1 crucial questions that need to be addressed for this entire topic to be clarified. Issue number 1, how to measure affect. Again, Bill Morgan said everything that needs to be said more than 20 years ago, and this statement remains unfortunately true today. Perhaps most of the literature dealing with the psychological effects of exercise has relied on the use of objective self-report inventories designed to measure constructs such as anxiety and depression. The extent to which these inventories can tap the psychometric domain of significance to the exerciser has not been evaluated. In other words, we've been using questionnaires of anxiety and depression to measure how people feel in response to exercise when we really don't know if those two states are the most relevant affective responses to exercise. As I said, the issue has persisted, and in 1993, Liz Govan and Larry Brawley said that the same thing. Because the affective experience that accompanies exercise has not been thoroughly described, a model of affect that has a wider breadth is more likely to capture the essence of exercise-induced affect than a model that at the outset limits the focus of investigation to specific emotions, namely anxiety, depression, and so on. So we need a model that's more broad, does not narrow our focus to just a few distinct states. So again, with Steve Petruzello in 2002, we proposed a transition to exactly such a model, the Effect Circumplex. The Effect Circumplex says that if you want to look at the global affective space, capture the entire domain of affect with as few dimensions as possible, you can do that with a two-dimensional model in which one dimension is affective valence that ranges from pleasure to displeasure, and the other is activation. If you combine these two dimensions, then you have one quadrant here, high activation pleasure, one quadrant here, high activation displeasure, low activation displeasure, low activation pleasure. For example, this is data from a factor analysis. These are some of the words that describe pleasant states, unpleasant states, high activation states, low activation states, high activation pleasant states, low activation unpleasant states, high activation unpleasant states, low activation pleasant states. And you see that if you do a factor analysis, and this comes out reliably, if you do a factor analysis, these states are going to be arranged in a circular fashion like that in exactly the pattern that you would expect based on the circumplex model. In addition to using words such as these, we can also tap directly the two underlying dimensions with one scale that measures valence, something that goes from I feel very good to I feel very bad, and one scale that measures perceived activation, something that goes from this is a low arousal state that I'm experiencing right now to a high arousal state. Moving to the second issue then, when should we measure affect? The traditional way has been to measure once before the exercise starts and then once or more times after the exercise ends. So if you do that, what you might find is that there might be a change, an improvement from pre to post of, say, 1 unit. The assumption that you're going to make in that case is that we move from point A to point B with a line, straight line. But how about if that wasn't the case, and what actually happened was that you moved from point A to point B with something like this, and there was, instead of a 1-point increase, there was a 6-point decrease during exercise. If you only measured pre and post, you would again misrepresent what the actual change in affect was. Issue number 3 is how to analyze the affective change at the level of group aggregates or at the level of individuals. In the case of affect, you can have bidirectional changes. In other words, you can have improvements in some individuals and then declines in other individuals. So if you have one trend going one way and another trend going another way, guess where the mean is going to go? Straight down the middle. So again, you would misrepresent the data. So we showed that in a study in 2000. This was a 30-minute stationary bike exercise at 60% of max. And this is the change at the level of the group aggregate during the session. No change in valence. But what actually happened when you looked at individuals is that approximately 50% of the sample showed a progressive decline, and about 50% of the sample showed a progressive improvement. So put everything together, and what do you get? A straight line, again, misrepresenting what the data is trying to show. Issue number 4 is how to standardize intensity. And there's a growing number of physiologists that reject, and have been doing so for years, the idea of percentages of max. Brian Wibb, for example, said that assigning work intensity either as multiples of resting metabolic rate or as percentages of VO2 max seems no longer justifiable. And the reason it seems no longer justifiable is that that practice ignores metabolic balance. So what they have proposed instead is a transition to a model that is based on the lactate threshold and the level of critical power. In this model, we have 3 domains of exercise intensity. One is moderate, that is, all the intensity is up to the lactate threshold, in which case most of the energy comes from aerobic metabolism and you can maintain steady state. The second intensity is from the lactate threshold to the level of critical power, in which case you can again establish steady state after a while, but at increased concentrations of lactate and VO2. And then the third domain is where you can no longer have a steady state, and VO2 and lactate and so on rise continuously until the point of exhaustion. So this makes more sense from an adaptational as well as a physiological standpoint. And the big one, the 5th question, what is the theoretical framework? That's actually a rhetorical question because there is no theoretical framework. The closest thing to a theoretical framework that we have is the inverted U, which says that basically if exercise intensity is too low, then exercise might be ineffective and not producing benefits. If the intensity is too high, it might be either ineffective or even aversive. So somewhere mid-range between those two extremes, you find optimal exercise intensities. Well, there are some problems with the inverted U. First of all, it doesn't account well for empirical findings, and there are several studies that go against this idea now. It is nomothetic. It assumes a stimulus-response effect, and it doesn't account for inter-individual variability, which is something that we see in our studies. And it's only descriptive because it doesn't tell you where those affective changes are coming from. So with all these things in mind, let's now look at some of the recent studies, those response studies. Wally Bixby did a study published in 2001 from the University of Maryland, and they compared 2 intensities. One was at 75% of the ventilatory threshold, and one was at the ventilatory threshold. If you look at the responses before the exercise started and the responses during recovery, there were no differences. So, of course, you would conclude that there are no dose-response effects. But if you take a look at the responses during, it's a whole different picture. Now you begin to see the effects of intensity. In the case of the lower intensity, the intensity at 75% of the ventilatory threshold, there was a significant improvement during. And when the intensity was started at the VT, then there was a significant decline during. So that's one study. We did something similar, but with a graded treadmill test, published in 2002. We used again the VT, the ventilatory threshold, which was assessed by such things as the deflection in the relationship between VCO2 and VO2, and the break point in the ventilatory equivalent for oxygen. These are standard methods for assessing the ventilatory threshold. And here's what happened. This is baseline. Exercise starts. And what you see up to the point of the ventilatory threshold is basically no change in valence. But once the ventilatory threshold is exceeded, then with every additional increase in work, you see declines, significant declines in valence. Then exercise stops, and within the first minute of cool down, this is what happens. There's an improvement in valence, a decrease in activation, and then as the cool down and recovery progress, you move into the familiar relaxing effect state produced by exercise. We replicated this, 2004, and we again confirmed that once you exceed the ventilatory threshold, that's precisely the point where you begin to see quadratic declines in how people feel. We extended this with, instead of a graded protocol, with a steady speed protocol, people exercising just below the VT, at the VT, or above the VT. And what you see here is that there are some declines, but they're not statistically significant when the intensity is below or at the ventilatory threshold. But when the intensity is just 10% of VO2 max above the ventilatory threshold, then the decline becomes significant from the 6th all the way to the 15th minute. Then some people have looked at, instead of ventilatory indices, they have looked at lactate indices. For example, Ed Acevedo, who is our chairman for today, looked at the onset of blood lactate accumulation. They showed that once the intensity exceeds the onset of blood lactate accumulation by 10% of VO2 max, two things happen. First of all, there is a statistically significant decline in how people feel. And second, we have a transition to negative values. People start to feel bad, not just worse than before. The same finding has been replicated. In this case, Acevedo's study was with runners, but in this case, it was a study with previously sedentary males. And they compared three conditions. One was above the lactate threshold, the other was below the lactate threshold, and the third was a self-selected intensity. These two lines show that the affective state was stable at the intensities that were either below the lactate threshold or self-selected intensity. But when the intensity was above the lactate threshold, again, we see two things: significant decline and a transition to actually negative values. How about correlates? What are those responses correlated with? Well, two clusters of variables. One is physiological variables, the other is cognitive variables. But the question is, does the strength of that relationship with affective responses vary as a function of intensity? In a study that's still unpublished, we found this is a relationship between self-efficacy, which is a cognitive variable, with the feeling scale, which is affective valence. And what you see is that the relationship was not significant when the intensity was low or when the intensity was very high, but it was significant around the ventilatory threshold. On the other hand, this is the relationship between the feeling scale, again, with the respiratory exchange ratio, physiological variable. What you find here is an increasingly more negative relationship. And it gets to pretty high values, almost close to 0.8, which for psychology, that's strong. If you combine these two in a multiple regression, what you find is that once you exceed the ventilatory threshold, then more and more of the variance in how people feel is explained by the physiological variable. Less and less variable is explained by the cognitive variable. So let's put the pieces of the puzzle together. We have proposed a model called the dual mode model. And this is a summary of it. It says that you can divide the spectrum of exercise intensity in 3 domains. First domain is below the ventilatory or lactate threshold, and in this domain you find homogeneity of responses, primarily pleasure, and low to moderate influence of cognitive factors. When the intensity is proximal to the ventilatory or lactate threshold, we find variability, with some people showing pleasure and some people showing displeasure. In this case, we have a strong relationship, strong influence of cognitive factors. When you go above the ventilatory lactate threshold, homogeneity arises again, the dominant response being displeasure, and we have a strong influence in this case of interoceptive factors. Now, the big question is this biologically viable? Well, before we actually get to that point, we have some obstacles ahead of us, and that is some people think that we shouldn't even be considering the question. Why? Well, Richard Lazarus said that neurophysiological concepts are generally inadequate templates for psychological concepts. So it's a futile enterprise to try and fit the psychological concepts into some kind of a neurophysiological model. The opposite view, which I'm biased, I admit it, this is what I favor, says the following. This is from Peter Lang. We need to temper behavioral and cognitive views of affect with what we know about the neurophysiology and neurochemical foundations of emotional behavior. This helps to limit inference and to keep our cognitive theories away from empty boxology. So with that addressed, let's move on and see if the model is biologically viable. My thinking in this area has been influenced a lot by Michel Cabanat. He said a few interesting things. Hedonic or affective responses to exercise have been shaped by evolution. They are linked to homeostatic perturbations, and pleasure signifies utility, whereas displeasure signifies danger. So what are the homeostatic perturbations that take place beyond the ventilatory and lactate threshold? Well, a bunch of things. Most of these are considered danger signals. The body has mechanisms specifically because it considers many of these changes to be threatening to homeostasis. There's accumulation of hydrogen ions, the pH begins to fall, there's development of metabolic acidosis, accelerated breakdown of the high-energy phosphates, inhibition of glycolysis and glycogenolysis and lipolysis, interference with the calcium triggering of muscle contractions. The oxygen cost of work increases, physiological steady state becomes difficult or in some cases impossible to maintain. There's a catecholamine threshold that happens very close to the VT or LT, and that has its own secondary effects. And then there's also evidence that the body enters a stress state because we see increases in CRH and say ACTH and cortisol. The HPA axis is activated. Now, what is needed for the dual mode model to be biologically viable? First of all, we need a system that collects and relays interoceptive information to the brain that is highly sensitive to intensity. We need a system that is hierarchically organized, and it includes both cortically and non-cortically mediated pathways. And we need appropriate gating mechanisms that can redirect interoceptive signals to the subcortical pathways when the intensity exceeds a critical threshold. We know that all this information, there is a system. It consists of the primary afferent neurons, the small diameter A, delta, and C neurons, and they collect all the information that is relevant. Information about mechanical stress, thermal stress, pH changes, hypoxia, and so on and so forth. Fernando Cervero has taught us what the system is that encodes the intensity of this information. And it consists of 3 different types of interoceptive neurons that are activated. One is called the low threshold, the second is called the high threshold, the third is called the silent. And these neurons are activated as the intensity of exercise, for example, or other interoceptive signals begins to increase. From Bud Craig, we have learned what happens in the spinal column and as this information ascends the spinal column. We have learned, for example, that there is a system in lamina 1 that is specifically activated in response to homeostatic perturbations. And we have also learned where those lamina 1 neurons terminate. They terminate in specific locations that are known. The nucleus of the solitary tract, the ventrolateral medulla, the parabrachial nucleus, and the periaqueductal gray. What happens then? Well, from this point on, we have to start speculating because there is no exercise research. So we're going to use other research that could be relevant. Joe Ledoux has revolutionized thinking in affective psychology by proposing that in addition to a high road of processing affective information, there is also an alternative low road that projects information directly from the thalamus to the amygdala, bypassing the cortex. So cognitive evaluation and the significance of stimuli is not necessary, according to Ledoux. And I'm going to skip this because I'm running out of time. Now, in exercise, we know that the amygdala is activated. Even though there are few studies, we know that the amygdala is activated in response to exercise. For example, this study showed a 56% increase in glucose utilization in the central nucleus. From other studies, we also know that in humans, the amygdala responds to exercise-relevant stimuli, such as increases and decreases in blood pressure, noxious somatic stimulation, induced dyspnea, and similar things in animals as well. So let's now see if we can extend Ledoux's model to make it relevant to interoceptive or exercise-related stimuli rather than exteroceptive stimuli that Ledoux was talking about, such as visual and auditory. The difference with exercise is that instead of one low road, we actually have multiple low roads. There is evidence that is going to be cited in the chapter that we're going to have in the psychobiology book, that there are projections to the amygdala from the periaqueductal gray, from the ventrolateral medulla, from the parabrachial nucleus, from the nucleus of the solitary tract, and even a direct projection from the spine. So there are multiple ways to stimulate the affective center of the brain without involving necessarily the cortex. In addition to this, we also have evidence that the appropriate gates are in place in the thalamus, the nucleus of the solitary tract, and the lateral nucleus of the amygdala, which could redirect the flow of information directly to the amygdala when the intensity is high enough to require this kind of immediate, direct affective response. So to summarize, we have the first emerging signs of a dose-response relationship between the intensity of exercise and affective responses. There is evidence that the role of cognitive and interoceptive correlates of affective responses is intensity dependent, with the interoceptive cues becoming dominant at higher intensities. And there is a possible neural basis for the interplay between cognitive and interoceptive factors, which at this point still awaits investigation. So, thank you very much.